Hey everyone! In today's video, we're going to take a look at the citric acid cycle. It's also named the TCA, which stands for tricarboxylic acid cycle. And it's also named after Hans Krebs. He's the scientist that won the Nobel Prize for fully describing this cycle. So it's also named the Krebs cycle. So all three names mean the exact same thing. And to start off, let's just recall from glycolysis that if we start with 1 mole or molecule of glucose, let's just say 1 mole, that after the process of glycolysis, we get 2 moles of pyruvate. So you're basically splitting the 6-carbon glucose into 2 3-carbon fragments called pyruvate. Now recall where this happens. This happens in the cytoplasm. Okay, but when oxygen is present in cells, now pyruvate is going to go through the PDC, the pyruvate dehydrogenase complex, which is in the inner mitochondrial membrane. So this is now going to go into the matrix and deposit acetyl-CoA, so 2 acetyl-CoAs. You also liberate some CO2. There are 2 CO2s that are released as gases, and we also have +2 NADHs. So the pyruvate dehydrogenase complex, that's again located in the inner mitochondrial membrane. So as the carbons pass through that, the acetyl-CoA units, this is called AC-CoA, abbreviate that, those are then deposited in the matrix, which is the innermost compartment of the mitochondria. So we'll just write mitochondria here. All right, so once those compounds now are deposited in the matrix, the 2 carbons of acetyl-CoA now are going to enter into what's called the Krebs cycle or the citric acid cycle. And ultimately, those 2 carbons are going to become completely oxidized so that carbon dioxide is then produced as the final product. So what we're going to now do is going to go through the detailed process of how one of these acetyl-CoA subunits goes through the Krebs cycle. Okay, so let's now go through the detailed process of the citric acid cycle. And again, recall that this is going to occur in the matrix of the mitochondria. So what I've drawn up here are the key compounds that are involved. And again, this is where we're going to start. Acetyl-CoA, which again came from the pyruvate dehydrogenase complex. We're going to just put the fact that this has 2 carbons. Okay, so that has 2 carbons. I'm going to go ahead and fill out the number of carbons for the rest of these compounds. So citrate is 5 carbons. I'm sorry, that has 6. 6. Isocitrate is 6 as well. Alpha-ketoglutarate is the only one with 5 carbons. Succinyl-CoA is going to have 4 carbons, so that's 4C. And then the remainder of this pathway is going to basically regenerate oxaloacetate. So this is 4 Cs, 4 Cs, and 4 Cs here. And oxaloacetate has 4 Cs. So in the process of the Krebs cycle, essentially what's happening now is we have a 2-carbon fragment of acetyl-CoA. It's going to combine with a 4-carbon fragment of oxaloacetate, and that's going to combine to make citrate. So that's going to be the very first step, and that's catalyzed by the enzyme called citrate synthase. Okay, citrate synthase is a heavily regulated enzyme. If it's turned off, essentially the Krebs cycle will shut down. So that key reaction now is going to basically get those carbons now involved in the Krebs cycle. And we talk about the Krebs cycle as being sort of a cyclic pathway, so we're going to go through what's called 1 turn of the so-called Krebs cycle here. All right, so let's just take a look at what's going on here. It's going to be difficult on this particular slide to show all the structures, so I do recommend that you would go through, draw this out on your paper, and then write in the detailed structures for each of these compounds. I'm just going to draw a couple of them. So oxaloacetate is a compound that you should know. It's going to have this structure to it. Okay, so it's a 4-carbon compound. It's got 2 carboxylic acids on either end. It has a ketone at carbon 2. Acetyl-CoA, we know the structure of that one. Okay, it's got the 2-carbon fragment there. So we're now going to combine those to form the molecule called citrate. So let me just draw that one out here. Okay, so that's the molecule called citrate. So what's happening here is that the methyl group of acetyl-CoA is reacting with the carbonyl carbon of oxaloacetate to convert this one into this hydroxyl. So envision now this right up here, I'll just put a little dotted line around it. That's coming from our acetyl-CoA. Okay, now one other thing that I should mention is that in this process, the CoA subunit is released. So the free sulfhydryl group of coenzyme A is released during that process. Okay, so now we have the 6-carbon compound called citrate, and you can see in this one there are 1, 2, 3 carboxylic acids. So that's one of the reasons this is called the citric acid cycle, but also the tricarboxylic acid cycle, because citrate has 3 carboxylic acids. Okay, so in the next step of this reaction, there's going to be essentially a dehydration and then a hydration. So we're going to remove water, and then water is going to go back on. So we're essentially going to flip-flop the position of this hydroxyl group. It's going to end up moving down to this position. I won't draw that structure just because I'll run out of room. And then lastly, or not lastly, the next step is going to be this key step where isocitrate is going to be converted to alpha-ketoglutarate. Now notice that this C6 molecule is going to be turned into a C5 molecule. So in this process, there's going to be a CO2 that's going to be released. And that step is essentially an irreversible step because the carbon dioxide is going to be liberated as a gas and essentially can't go backwards. All right, so that enzyme is a critical regulatory enzyme as well. It's called Isocitrate Dehydrogenase. We'll put that in kind of a cloud box there. And one of the key molecules that's produced by isocitrate dehydrogenase is going to be NADH. All right, so we'll just show that coming out here. There's going to be an NADH produced there. You may recall that any enzyme that has the word dehydrogenase in its name typically is going to use the NAD cofactor. So we would have NAD plus going into this reaction, and then out would come NADH. So isocitrate dehydrogenase, it's basically going to chop down the 6-carbon isocitrate into the 5-carbon alpha-ketoglutarate. And since I do have space here, I'm going to go ahead and just draw this compound out. All right, so that's the molecule called alpha-ketoglutarate. All right, the very next step is going to be another decarboxylation steps, because notice we're going to go from a C5 alpha-ketoglutarate to a C4 succinyl-CoA. So there's going to be another CO2 that's going to be liberated. And we're going to now make succinyl-CoA. And the enzyme that's going to do that, not surprisingly, is called alpha-ketoglutarate dehydrogenase. I'll write that down here. Okay, and that enzyme is again going to require an NAD+, and that will convert it to an NADH. I'm going to put that in a box. So these two enzymes then, the isocitrate dehydrogenase and the alpha-ketoglutarate dehydrogenase, they're critically important regulatory enzymes in this pathway. And they are essentially performing an irreversible reaction. So once the CO2 comes off of each of those, it really can't go backwards. And along the way, there are two equivalents of NADH that are then produced. Okay, so we now have a 4-carbon succinyl-CoA. I won't draw out the structure of this. Oh, by the way, an HS-CoA subunit had to come in here. It's going to get added on to the succinyl group. In this next pathway, or part of the pathway, succinyl-CoA, a C4 compound, is now going to be sort of recycled back up to the 4-carbon oxaloacetate through a series of enzymatic steps. And let's just talk about what's going on in these two steps. In this one, the succinyl-CoA going to succinate step, this is going to require a GTP molecule, a GDP molecule, and that's going to be converted to a GTP. Now, energetically, we're going to consider that to be equivalent to an ATP molecule. That step is called, or is catalyzed by succinyl-CoA synthetase. So then now we have succinate, which is a C4 carbon, and we're now going to convert that succinate into fumarate. Now, over on the side, I'm going to go ahead and draw the structures of succinate. And then I'll write fumarate as well. I know I'm kind of drawing over my face here. It may be kind of hard to see, but it's the best I can do with the space I've got. All right, so notice what's happening in this step. A CH2-CH2 bond, so a carbon-carbon single bond, is being converted into a carbon-carbon double bond. And in order to do that, this requires a special cofactor. So FAD is going to be converted to FADH2. So these two hydrogens here and here are going to be added to the FAD cofactor to make FADH2. All right, and not surprisingly, the name of this enzyme is called Succinate Dehydrogenase. So it's taking succinate and dehydrogenating it. That also is a key regulatory enzyme. Okay, last two steps here of the pathway. Malate is going to be produced, so we're going to add water to this. And we're going to essentially be adding the oxygen to the 2-carbon of fumarate. So let me go ahead and draw the structure of malate here. Okay, so we've essentially added water. I'll just put a little dashed line around the OH and the H there. That's now going to make this compound called malate. And then lastly, to convert malate to oxaloacetate, the enzyme called Malate Dehydrogenase is going to now use an NAD+, and that's going to make another NADH. So obviously, a lot is going on in this figure. I would highly recommend that you sit down with a clean sheet of paper, redraw this out, and pay close attention to the enzymes that are making NADH. So there's isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase. There's also this one here, malate dehydrogenase. I sort of don't have room, I'll just write it here. Mal, DEH. Yeah, that's malate dehydrogenase. Those three enzymes that produce NADH are key regulatory enzymes, as is citrate synthase. That's the one that starts the whole process off. And then lastly, succinate dehydrogenase, which performs the FAD to FADH2 conversion. That also is a regulatory enzyme. So pay close attention to those. On the next slide, I'm just going to summarize the main products of this to kind of simplify this a little bit for you. But definitely, you're going to want to get to know this figure fairly intricately. There is a lot going on here, and it's going to take some practice. All right, so to summarize the citric acid cycle, let's just list out the key products that are produced from one turn of the citric acid cycle. So we have one acetyl-CoA, okay, and what that will then produce will be the following. It's going to produce 3 NADH, 1 FADH2, one GTP, which I'm going to put in parentheses ATP because that's equivalent to an ATP. Okay, and then as byproducts from this, we'll also get minus 2 CO2 molecules. All right, so that would be for one turn. Okay, however, if we trace this all the way back to glucose, recall that glucose ultimately gave rise to 2 acetyl-CoAs. So in order for the carbons of glucose to be completely oxidized, we have to do 2 turns. So we're going to just list out 2 turns. And ultimately, we'll then just take these numbers and multiply each of them by 2. So by going through 2 turns of the Krebs cycle, we're ultimately going to get 6 NADHs, 2 FADH2s, 2 GTP/ATP, and then minus 4 CO2s. All right. Now, let's also add above this, recall that in order for the pyruvate to go through the pyruvate dehydrogenase complex, that produced an NADH plus CO2. So ultimately, from PDC, from the pyruvate dehydrogenase complex, because we have 2 acetyl-CoA subunits going through that, or being produced, we're going to add on an additional 2 NADH from that step. And then there would also be minus 2 CO2s. So here we have a total of minus 6 CO2s. That represents ultimately the 6 carbons from glucose that have now become completely oxidized. So for the whole process then, once we start getting into the, again, all of this is happening in the mitochondria, we're ultimately making a total of 8 NADHs, 2 FADH2s, and 2 GTP/ATPs. Now again, all of this is happening in the matrix, and in one of the subsequent videos we're going to talk about now what these NADH and FADH2s actually do. They are electron carriers. They're going to interact with what's called the electron transport chain, and ultimately make a lot of ATP. All right, so that summarizes now the main products then of the Krebs cycle. Definitely go back, get a clean sheet of paper, write out the full cycle so you can understand all the various steps. And good luck with everything.